Hello everyone, we are Group 5. Today we are going to present a paper from Nature Biotechnology called An Engineered Influenza Virus Deliver Antigens for Lung Cancer Vaccination. Author is Professor Zhou from China. Professor Zhou is with Peking University and Shenzhen Bay Laboratory. Research focuses on immunotherapy, especially drug development. One of his previous studies talking about a new form of vaccine. They use influenza A virus as particle, use gene editing to treat the virus, which make vaccine less toxicity to body. So what about this paper? Now they continue previous experiment, use this vaccine on animal model and make sure it's effective. So let's talk about vaccine. In this paper, they want to generate a live but non-productive influenza A virus, or short in IAV, to improve safety of IAV vaccine. First, they modify IAV by introduce 4 premature termination codon that will inhibit virus replication. Only when adding unnatural amino acid with orthogonal tRNA will lead to virus production, and they call this kind of live attenuated IAV virus as PTC. Second, they insert antigen peptide and CpG as adjuvant on vaccine surface. It can increase cell uptake and enhance immune response. This modified virus called peptide-armed PTC virus, or short in PAPV. At last, insert anti-PD-L1 gene into virus against immunosuppressive environment. This kind of vaccine called PAPV-ICI, which means immune checkpoint inhibitor. This is the vaccine they use. So let's go to the result part.
They first observe the morphology of different IAV. WSN is wild type, PTC is attenuated IAV, and PAPV is peptide-modified PTC. As we can see in Figure B, it is almost identical between WSN, PTC, and PAPV. As for infectivity test, cell viability is used as an evaluation to indirectly measure the virus infection and replication. We can see that PTC and PAPV will infect cells only in the presence of UAA, confirming the safety of PAPV for the host. Next, they test the in vivo toxicity of PAPV to C57 mice by measuring the body weight and pulmonary edema after intranasal administration. As we can see, PAPV showed no toxicity for treatment in C57 mice. They further assess whether non-productive virus exert anti-tumor function or not. They generate non-metastatic melanoma model in C57 mice by intravenous injection of tumor cells. IAV are intranasal injected into mice after 1 week tumor implantation. The result is shown in Figure F. PAPV significantly reduced the number of melanoma foci in the lung, suggesting that PAPV can promote anti-tumor efficacy. Next, they investigate the capacity of PAPV in facilitating tissue distribution and uptake. The AG1 and CpG are labeled with FITC and Cy3 fluorescent respectively. Free AG1, CpG, or PAPV is administered intranasally into mice and circulation for 24 hours and 48 hours. According to the result, we can see that peptide tethered on virus enhance the antigen uptake and accumulation in lung tissue. Because dendritic cells serve as an important role in antigen presentation and immune response, they further evaluate the ability of PAPV in cellular uptake and presentation of AG1 in dendritic cell. We can see that free AG1 and CpG only show very poor signal in antigen presentation. On the other hand, PAPV exert robust FITC signal on cell membrane, indicating that PAPV significantly enhance the capacity of antigen presentation in DC. Moreover, dendritic cell activation is required for activation of T cell. They also examine the DC activation marker by flow cytometry. CD80 and CD86 are DC activation marker. MSC2 is antigen presenting marker. We can see that HU1 alone cannot induce DC activation. PTC or CpG alone lead to around two-fold improvement. The PTC virus pretreated with CpG group and PAPV group induce the highest capacity of DC activation. Coincident with the upregulating markers on the activated DC, pro-inflammatory cytokines were found to be secreted from both PTC pretreated with CpG group and PAPV group. So sum up of previous result, we can conclude that PAPV act as superior driver that improve antigen uptake to DC, promote antigen duration and presentation, as well as facilitate DC activation.
Next, they examine the effect of intranasal injection of PAPV in metastatic lung melanoma. In Figure A, we can see that PAPV increased the antigen expression on DCs. And in Figure B, the PAPV treatment showed an increase of the immune cell amounts in the metastatic lung, including CD4 positive T cell, CD8 positive T cell, NK cells, and antigen-presenting dendritic cells. And in Figure C, the PAPV treatment shows the increase of the antigen-specific CD8 positive T cell numbers, with an effector function represented by the interferon gamma and TNF-alpha here. And then they want to determine the anti-tumor efficacy of the PAPV in the mouse model of lung metastatic melanoma. So the treatment timeline was shown here that after 2 injections of the PAPV, the mice was intravenously injected by the B16-OVA cells. And the result was shown here that the PAPV treatment resulted in the reduction of the number of tumor foci here, with an increase of the CD8 positive T cells with an antigen specific. And in Figure F, the PAPV treatment shows to increase the antibody IgG titer here after the first treatment and increase more after the second treatment. The PAPV also stimulated mucosal immunity represented by the IgA titer here. Next, they want to evaluate whether PAPV immunization could promote an inflammatory response in immunosuppressive tumor microenvironments. So the mice were intravenously inoculated with B16F10 OVA cells first, and followed by 2 intranasal immunization with PAPV, or the antigen and the adjuvants alone after 14 days. So in Figure G here, we can see that the anti-tumor immune cells, including CD4, CD8, NKs, DCs, and the M1 macrophages are all increased after the PAPV treatments. And the pro-tumor type immune cells, including the myeloid-derived suppressor cells, Tregs, M2 macrophages, are all decreased after PAPV treatments. And in Figure H, the PAPV treatment shows an increase of the CD8 positive T cell and CD4 positive T cell infiltrating into the lung area. And given that memory T cells play a critical role in prolonged protection against cancer, so they next tested whether PAPV treatment generated memory T cells by adoptive transferring of the splenocytes. And we can see that in Figure I, transferred splenocyte and injected the tumor cells. It resulted in the tumor inhibition. And if we implanted tumor cell first and dotted transfer the splenocyte, we can see the delay of the tumor growth. In Figure J, the PAPV treatment shows to increase the number of the resident memory T cells after the 50 days treatments and the 100 days treatments. So these results show that the PAPV indeed could increase the inflammatory response in the immunosuppressive tumor microenvironment with an memory response after the injection. Next, they engineered the PAPV by insertion of a gene fragment encoding the anti-PD-L1 antibody. So they want to test the synergistic effect of the PAPV with anti-PD-L1 antibody in the lung metastatic model. So the treatment timeline was shown in the Figure C here. After 7 days of the tumor inoculation, the mice were treated with 3 doses of the different treatment groups. And we can see the results here that the PAPV combined with the immune checkpoint inhibitor, the anti-PD-L1 antibody, could inhibit the tumor growth in the lung area and prolong the overall mice survival. And this result comes from the increase of the activation of the CD8 positive T cell, which represented by the interferon gamma and granzyme B here. These two markers are increased after a PAPV treatment. And the PAPV also increased the TH1 subtypes. In figure E, the immunohistochemistry revealed that the significant increase was in the infiltrating CD4 positive and CD8 positive T cells in the metastatic bearing lungs after the treatments. They also conduct a tumor rechallenge study that they will injected the tumor cells in the cured mice in the previous experiments to test the immune memory after the PAPV treatments. And we found that after the tumor rechallenge, the PAPV combined with the immune checkpoint inhibitor could result in the inhibition of tumor growth and to prolong the mice survival here. Finally, they want to evaluate the potential of this strategy against other cancer. So the CD26 colon cancer model and the 41 breast cancer model was chosen here, and the PAPV combined with the immune checkpoint inhibitor inhibit the lung tumor growth in both models and increased the overall mice survival in both models.
Here's a brief summary. The PTC virus infection can turn on the immune response in the tumor microenvironment, and the PAPV endow high DC recruitment that caused the increase of T cell population and activation. Furthermore, combined with PD-L1 nanobody can prolong the immune response. This viral-based vaccine can enhance mucosal immunity successfully. It's a breakthrough, and it can also induce a long-term protective T cell memory, but there's still some limitation. We need to know what the specific antigen is, and the author didn't test any human cancer cells and in a human immune environment. The last point is, intranasal is not the clinical route of administration. They need to develop a novel aerosol inhalation aid that is usually hard to use correctly by patient. Thank you for listening.